Welcome back, everyone. First, let me know if audio and video both are working fine. Okay, wonderful. So, let's move on with the chapter of biotechnology. The very first thing that we need to understand in this entire chapter is the basic meaning of what is DNA, what are genes, and what are chromosomes. And these are terms that will keep coming across in this chapter. So, that's why it becomes essential that you understand the basic meaning of what these are. Now, when you think of any organism, whether it's a microorganism or a plant or an animal or humans, any organism, there would be various organ systems, right? In particular, let's say when we talk about humans, you can say that we have digestive system, nervous system, muscular system, skeleton system. All these are various organ systems that we have in our body, which are made of what? Which are made of various organs. They're all made of various organs. Organs like, for example, heart is an organ, brain is an organ, kidney is an organ, liver is an organ. All these are organs in our body. Now, all these organs, basically organs are nothing but set of tissues. They are set of tissues which perform a specific function. So all these organs, they are made of tissues, and these tissues are made of cells, right? They are made of cells. Now, when it comes to any organ or any organism, they're all made of basic cells. Now, cells, what we say is that cells are basic structural and functional unit of life. Structural and functional unit of life. Now, when we say structural and functional, what is the meaning of this word structural and functional? Any cell in itself provides for all the different types of functions that are required to be done. And in a manner, you can say that the cell in itself is a complete organism. There are many organisms which are unicellular organisms, right? Where you have that organism made of just one cell, right? They're just made of one cell. And in that one cell, they are able to survive because various uh, functions that are required for their survival, it's all there in that one cell. So that's why they are unicellular organisms. Similarly, in multicellular organisms, there are various different types of cells, and all these cells have their own various functions. Now, when it comes to all these cells, we know that inside the cell there are various organelles, right? There are various cell organelles, and all these cell organelles, they have their own specific function. For example, you might have heard of a mitochondria. Now, mitochondria inside the cell is also called as the powerhouse of the cell. This you might remember, that it's called as the powerhouse of the cell. Now, why we call it as powerhouse of the cell is mainly because it's something that provides energy to the cell. So every cellular activity happening inside the cell or by the cell, the energy that it gets is from mitochondria. So that's why powerhouse of the cell. So similarly, there are other organelles, and each and every organelle has their own function. Now here, when we go inside the cell, there is a very, very important part of the cell, which is called as the nucleus. We have a very important part of the cell, which is called as the nucleus. Now, in case of a nucleus, what we understand is that nucleus also is called as the brain of the cell. And why we call it as the brain of the cell is because all the activities being done by the cell, they're all controlled by the nucleus. So it is related to the control and coordination of all the activities which are happening at cellular level. So that's why various different things that the cell does, each and every function of the cell, there is the nucleus that also controls and coordinates for all these activities inside the cell. Now, in the nucleus, we have thread like structures which are called as chromosomes. We have thread like structures which are called as chromosomes inside the nucleus. Now, these chromosomes, for example, in case of humans, you might know that we have 23 pairs of chromosomes. If you look at this structure, this is where we are now. We have a cell. Inside the cell, we have a nucleus. And inside the nucleus, we have small thread-like structures, which are called as chromosomes. Now, these chromosomes basically are made entirely of something called as DNA. But there are segments of DNA which make up a gene. There are segments of DNA which make up the gene. So in this scenario, you can say that DNA is the smallest. Now DNA, a bunch of DNA together will make up a gene, and there are sets of genes that you have all across the cell, and that makes up the chromosome. So this is where you can say that this is inside the cell. This is what you see inside the cell. Now outside the cell we have various organs, organelles, organism. Ultimately we come to an organism, and these organisms, they make up communities. Sorry, first they will make up populations, and populations will make up communities. So there are populations of organism, right? So any organism, first there will be population of that organism, and when different organisms live together, this is what is called as community. And these communities together will be called as ecosystem. They will make up the ecosystem. So overall this is how we see, and ultimately you can say that it makes up biome or biosphere. So in a manner we see that these are the various levels of organization. One is the ecological level of organization, then you have the biological level of organism, and then organization, and then you have the cellular level of organization. So in this manner there are various different kinds of organization that we see. We are concerned about this organization which is inside the nucleus. Now overall, when we see that, when we talk about a DNA basically contains all the instructions, that are all the characteristics, everything that is required for having specific characters. Now, when it comes to, let's say, uh, chromosomes, DNA, and genes, understand the basic difference between all these three terms. First and foremost, understand this, that DNA is the smallest here. Genes are made of segments of DNA, and the entire all these segments together, they will make up the chromosome. So this is how we look at the various aspects. So overall, just understand that all the instructions that are required for building of an organism, each and every character of an organism, all the instructions are there inside the DNA. So that's why they become very, very crucial, because everything that is required to, every instruction that is required to build and maintain an organism, it's all there in the DNA. So that's where DNA from that perspective becomes very, very important. And then when it comes to genes, genes are made of segments of DNA. A segment of DNA that has a particular character, a particular property that any organism has. It's all about of genes. It's all about it's all because of genes, and these genes are made of DNA. Let me give you an example. If you think of any specific character that everybody around you has, suppose that we are talking about only humans, so you might see that color of skin in humans is different, height of the humans are different, their metabolic activities are different, color of the eye would be different, each and color of the hair is different when you look at a larger sample. So each and everything is a character, and all these characters, they come from genes. All right. So there would be a gene that is determining the color of the eye. There is a gene that is determining what is the color of the skin. There is a gene that is responsible for what's the growth going to be like in that individual. So everything there, for every character that you see, for every trait that you see, there is a gene that is leading to that character. So that's why genes basically, they are made of a bunch of DNA, a segment of DNA which is responsible for a particular character or a particular trait. So this is how we look at the level of organization between the DNA and the gene. So in case of DNA, we say that it is deoxyribonucleic acid, and it is made of, we'll discuss about that later, it is made of chemical bases, and all these bases together will make up the DNA ultimately. So that's why DNA, genes are nothing but made of specific segment of DNA, and hence all the specific traits and characters of an individual are because of the particular genes that they have. Now overall, these genes, they will make up, all these genes together, they will make up the chromosome. All right, they'll make up the chromosome. and there are a total of 23 pairs of chromosomes. And if you just think of the numbers also, these numbers will tell you about what is big and what is small. So for example, when you uh, look at the number of chromosomes, there are 23 pairs of chromosomes that a human body has. So 23 pairs, meaning that there are a total of 46 chromosomes in a human body. 46 chromosomes, meaning 23 pairs of chromosomes, out of which 22 pairs out of which 22 pairs are similar in males and females, and the 23rd pair is something that determines whether it will be a male or a female. So the 23rd pair is different. All the other pairs of chromosomes, they are the same. So in total there are 46 chromosomes, meaning 23 pairs of chromosomes. The number of genes in the human body ranges between 20 to 25,000. A lot of studies have been done on it. Initially there was something called as the Human Genome Project, and Human Genome Project estimated the number of genes to be around 40,000 initially. But then with all the studies and research that went into studying of the genes, later it was determined that the number might be in the range of 20 to 25,000. So 23 pairs of chromosomes, 20 to 25,000 numbers of genes. And when it comes to DNA and the number of DNA, uh, calculating the number of DNA becomes almost impossible, because every cell in the body, you can say that every cell in the body, there could be more than 3 billion base pairs that could be present in every cell. I mean every nucleus of a particular cell. And there are so many cells in our body, and every cell has an estimate of, I think, 3.2 billion pairs of DNA. So that's why it becomes impossible for us to even think of how exactly how much would be the number of DNA. So that's why I'm saying that the DNA is the smallest. Segments of DNA together will make up the gene, and all these genes will ultimately, this is where you have the chromosomes. So so that's why all these things, they become important. So in case of DNA, what we can simply say is that when it comes to a DNA contains, its, you can say in a manner, uh, blueprint. It's like a of the instructions blueprint of the instructions that contain all the information all the information needed to build and maintain an organism. All right, so this is how we can define what DNA is. And then after that, when it comes to genes, so genes, in a manner, you can say that these are segments of DNA. These are segments of DNA responsible for a certain trait. Right, segment of DNA that is uh, responsible for a certain trait. And then you have chromosomes. So chromosomes basically are thread like structures. All right, these are thread like structures where you can say that they are organized structures. These are organized structure of DNA. All right, these are organized structure of DNA. So in this manner, this is how we understand about all of them. Now, so that's why when you look at chromosomes, we know that these are long, coiled up, thread-like structures made of DNA and proteins, and carries all the genetic information in the form of genes. And as we discussed, that humans have a total of 23 pairs of chromosomes altogether. In case of genes, What we discussed is that these are made of segments of DNA, and they have the instructions for the development and the functions of the living organisms. All the particular traits, as I told you, that whether it's about the color of the hair or color of the skin, the height, weight, everything, how many hair will be left on the scalp, it's all the genes. It's all there in the genes, and the genes will decide a lot of these things. So that's why genes carry all the specific characters, and that's why these become the basic units of heredity, and these are passed on from generation to generation. That uh, birth of a new individual, whenever the birth of new individual happens, the genes will be carried from the mother and father. There will be a mixing of genes from the mother and father, and hence a set of genes will be present, and that's why there will be some characters that will be inherited from the father, and there will be some characters that will be inherited from the mother. So in this case we see that this is something that works in terms of genes. Now, when it comes to DNA, we are talking about a molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all living organisms. Now, when it comes to, let's say, a DNA, DNA is made of four pairs, base pairs, which are called as adenine, thymine, guanine, and cytosine, where adenine and thymine always combine together, and guanine and cytosine always combine together, and they would create different types of DNA, different pairs of DNA. So for example, AT can combine with GC, so that becomes one. AT can combine with CG, then TA can combine with CG, or TA can combine with GC, and all these kind of combinations are possible. All right. So all these combinations are possible, and all these combinations together, when they come in a particular form, then they will form a gene. If they make sense, they will form a gene. So this is how exactly the things are done. So overall, when it comes to the DNA, this is basically what we understand about the DNA. Now understand one more thing, that when it comes to the DNA, the structure that you see is called as the the double helical structure, and as you can see, it's like a twisted ladder. That if you have a ladder and you twist that ladder, the kind of structure that you will get is the structure of the DNA. So that is basically where we understand the DNA, and in this particular scenario. So overall, that's why when you look at DNA, genes and chromosomes, just think of a book. All right. If suppose chromosomes, if let's say chromosomes together make up all the any book, then all the chapters that you have, the chapters are the 23 pairs of chromosomes. So you can say that in a manner you have a book which has 23 chapters, and all these 23 chapters are the 23 pairs of chromosomes. All right. Then below this, the next level is of genes. So genes, you can say that in a manner, are the sentences that you have in any given chapter, or the words that, let's say, the words that you have in a given chapter, those words are the genes. All right, the words that you have in every chapter are the genes, and they are made of letters, and these letters are like DNA. So when the letters combine in a logical form, they create a word. Similarly, when DNA combine in a logical form, they create a gene. So all these letters are like DNA. So let's say if you have A, B, and C with you, all right. Let's say if you have three letters A, B, and C, when they combine as CAB, they make sense. So this is similarly what happens with in the case of a DNA as well. That when DNA combine in a logical form, they can create a gene which is responsible for a particular trait. So this is how we have to understand in the scenario of how DNA functions. Now, one more uh, concept related to the DNA is the concept of RNA, ribonucleic acid. In case of DNA, we say that it is deoxyribonucleic acid, and RNA is ribonucleic acid. Now, in case of RNA, RNA contains the instructions for making up proteins. RNA will have instructions which is helpful in making proteins. It contains all the instructions that are required for making proteins in the body. And that's why it is in a manner you can say that it is like a messenger. It's like a messenger that is carrying all the instructions. So it would be contacting, or it will be in contact with a segment of DNA. It will carry all the information, and it will help in making of proteins or synthesizing proteins in the body. So that's why they become very important in that sense. So from prelims perspective, it's also important for us to understand the basics of even DNA and RNA and how they are related to one another. So when it comes to DNA and RNA, first and foremost, structurally they are different. And how exactly the structure is different? If you see, DNA has a double stranded molecule, whereas RNA is a single stranded molecule. So that's why RNA is like this. This is the structure of RNA, and DNA, as we discussed, that this is the structure of DNA. So that's why the basic difference comes, where we are saying that DNA is like a double helical structure or a twisted ladder kind of structure, and half of that ladder is what you have as RNA. Now, although both are instruction manuals in a manner, but when it comes to DNA, we know that DNA will be the one which will transfer the genetic information, and hence it will carry all the specific characters with regards to the function of the body. And when it comes to the RNA, in case of RNA, we see that this is where it contains the code that is required for protein production from the nucleus to the ribosomes. Now, at the same time, another difference is also in the base pairs that they have. So in case of DNA, what we discuss is that you have guanine, thiamine, cytosine, and adenine. Whereas in case of RNA, all the other three are the same, but instead of thiamine, you have uracil. So guanine, adenine, and cytosine are the same, but uracil is there instead of thiamine. So that's a basic difference that we have between DNA and RNA. Again, from the perspective of the location of where they are located, DNA, we know that they are inside the nucleus, because inside the nucleus you have chromosomes. Inside the chromosomes you have segments of DNA that make up the gene. So that's why they are placed inside the nucleus. Whereas when it comes to RNA, RNA is placed outside the nucleus also. It's there in the cytoplasm, it's there everywhere, because it will carry the instructions which are required for making proteins for different parts of the cell. So that's why the cytoplasm, nucleus, and ribosomes, all of it, they contain RNA. Whereas when it comes to DNA, it is largely found inside the nucleus, but not only inside the nucleus, it is also found in mitochondria, and in plants, it is also found in plastids. They're also found in plastids. So plastids are there in the plants, and these plastids also contain DNA. But the important DNA, the one that carries all the important information, or all the characters, all the specific characters of any individual, they are all there in the nucleus. Outside the nucleus, the other kind of DNA that we have, they are not specific in character. All right, they are not specific in character. So for example, when I talked about color of the eye, color of the hair, color of the skin, etc., all these specific characters are there inside the DNA in the nucleus. Whereas in the mitochondria, the DNA that we have, they are common DNA, the DNA that are common across the human species. That, for example, how the heart functions, how the basic body functions are, how the blood would go from one place to another, all these kind of things, which are, you can say, general information about human race or human species, they will be there in the DNA. Sorry, in the mitochondrial DNA. Whereas when it comes to the DNA inside the nucleus, they will be responsible for all the specific characters. So this is how we see the difference between the DNA and the RNA. So that's why just understand some of the basics that we are discussing. That DNA basically becomes the basic building block. Genes are the segments of DNA, and chromosomes are the house of the genes. Basically, they are containing everything that you have. So that's why you have to understand the basics and how they are different from one another. And that's why I told you that even if you see in terms of numbers, the numbers are pretty different. That the number of chromosomes, for example, as we discussed, that the number of chromosomes, we have 46 in total, that is 23. pairs, right? We have 23 pairs. And then after this, when we talk about the genes, in terms of genes, what we have discussed is that there are possible 20 to 20,000, 25,000 genes in the human body. 20,000 to 25,000. And in terms of DNA, there are 3.2 billion pairs per cell. So that's why this is a big number when we talk about the DNA. Any question if you have anything to ask from what we discussed until now? By pairs of DNA, we mean two strands. Uh, Not exactly. So basically, when it comes to, see, let me just go back to DNA. So when we say pair here, this is what a base pair is. That when these two combine together, they basically make up a pair. All right. So this is what we talk about as the base pairs in case of DNA. For non-science students, won't it be tough to remember all the minute details? So see, you only have to remember the details which are there on the screen. Beyond the screen, whatever stories I'm telling you, they are not important. They are just additional things that I'm telling you. When you revise, the only things that are important are the things that are there in the slides. Mitochondrial DNA is different. So that's what I told you, that mitochondrial DNA is different because it does not carry the specific characters, but rather the species level characters. 23 pairs of chromosomes per cell, right? That's correct. That's correct. What is genetic code? So genetic code, basically, when we are talking about, it's nothing but, you can say, the number of genes that you have and all the genes that are responsible for a particular trait, how you look at all those things together. Can you please explain base pairs? So base pairs, basically, base, if you remember, there is something called as an acid and a base, right? So that base is what we are talking about, that these are bases that we have, and these two, uh, these bases, they combine together, they become the basic, you can say, in a manner, they become the memory unit for you. I mean, just like how for your mobile phones or your laptops or your computers, you might have some hard disk, and all that, those hard disks are uh, having all the storing all the information. So all these information, the basic unit of information is in these base pairs, and they make up the DNA, the molecules of DNA. Is DNA change possible? See, the genes that we have in our body, they do change, they do modify, they because we might be born with a certain set of genes which are related to what we have inherited from our mothers and fathers, but over a period of time, due to external conditions, environmental conditions, and everything with regards to that individual, there are changes and modifications that do happen in the genes. So yes, this is possible. All right, just give me 2 minutes, please. Yeah. So now coming to biotechnology, and when it comes to this chapter, what we are basically studying is all the various applications of biotechnology. This is what the crux of this chapter is, to study the applications of biotechnology. And for that, we classify biotechnology into various branches. Now, when we look at the branches of biotechnology, there is a way in which this classification is done where we color code the branches, where we are saying that green biotechnology is about agriculture, red biotechnology is about medical applications, white biotechnology is industrial applications, blue biotechnology is something which is related to water or water related applications. Then you have gray biotechnology for environment, you have brown biotechnology for anything that you do in the deserts or arid regions. So that's why for each and every area, or for each and every branch, or wherever we are applying biotechnology in one way or the other, there is a color that we assign to every one of the branches. So that's why green biotechnology when applied to agriculture, red biotechnology when applied to medical science, blue biotechnology related to water, white biotechnology related to industrial processes, gray biotechnology related to the environment, brown biotechnology related to arid zones or deserts. Then we have also violet biotechnology. Now, violet biotechnology basically is about uh, everything else about biotechnology: the laws, the ethics, the philosophical issues, the documents, the technical papers, the intellectual property rights. All these things related to biotechnology, they will be counted as violet biotechnology. All right. So violet biotechnology, and then lastly we have something called as dark biotechnology. Now dark biotechnology is, you can say in a manner, misuse of biotechnology. When biotechnology is not being used for something fruitful, then in that case it is dark biotechnology. So bioterrorism, biowarfare, uh, any application where you are not using it for the benefit or something that is going to help the society altogether, then in that case it will be called as dark biotechnology. So we'll discuss about all these in detail. To start with, we are starting with the medical applications of biotechnology. Now, in case of medical applications, understand the meaning again, that when we say medical applications, we are talking about anything that can be useful, whether as a treatment, or maybe as in case of medicines, or maybe a medical therapy that we can make out of it. So, in all these cases, these will be called as medical applications of biotechnology. Now, when it comes to the medical applications, there one thing that you must also know is, most, I mean, out of all the investment that happens in the field of biotechnology, more than 70% of the investment happens only in medical biotechnology or red biotechnology. That, if you look at the data for India, out of 100 rupees that in India, out of 100 rupees that is spent on biotechnology, more than 70 rupees is spent only on biopharmaceutics, which is a form of red biotechnology. So that's why I said that more than 70% of the money that flows in biotechnology flows in medical applications. And also because of a reason, because this is where biotechnology gives us also a chance to solve the unsolvable, for all the diseases that we haven't been able to find cure for, or we haven't been able to find even a vaccine for. In all these cases, this is where we see that medical biotechnology becomes very, very crucial, because this is where uh, we see that until now, whatever has been causing a lot of issues for us, what all the problems, health related issues, all these things can be solved with the help of biotechnology. That's why the first topic that we have right here is gene therapy. Now, in case of gene therapy, what we are saying is that this is the therapeutic delivery of nucleic acid into a patient's cell as a drug to treat a disease. Now, when it comes to gene therapy, it's largely about how we can make the use of genes. Now, what we understand about genes is that genes carry particular characters, right? Particular characteristics. Now, when we say therapeutic delivery, therapeutic is anything medical in nature, all right? So, anything related to medical treatment or anything related to uh, healthcare. So, therapeutic, that's why the term therapeutic is used there. So, now what we are saying is that here we are going to use genes to our advantage. What we know about genes until now is that genes are associated with a particular property, that for every character, for every trait that an individual has, there is a gene behind it. So sometimes absence of a gene can be a problem, that there was a gene which was related to a certain character, and now that this gene is absent in the body, it also means that it will lead to that character not being present, or sometimes it can lead to a disease. Absence of a gene can lead to a disease. Changes in the gene, or let's say some kind of modifications have happened in the genes, they will also lead to uh, they will also lead to some kind of disease. Or maybe there is a gene that is causing all the trouble. Maybe there is a gene that is causing the problem. So that's why sometimes presence of an unwanted gene also could be an issue. So that's why when we apply gene therapy could be about either addition of a gene or removal of a gene, or it could be about modification of a gene, or maybe even replacement of a gene. So these are the things that we'll try to do when it comes to gene therapy. That either we will try to add a gene which is absent from the body, or we will remove a gene which is causing the trouble in the body, or we will try to modify the gene in which some kind of problems have occurred, or maybe we'll replace the gene where the issues have happened. Correct? So these are the ways in which genes can be used to solve a problem. Now today we see that for a lot of such diseases where we did not have a medical therapy, we did not have a medicine, we did not have a vaccine, in all these cases gene therapy has come out to be the savior. And we have seen that there are cases where gene therapy has been able to solve some of these issues which were unsolvable otherwise. So that's why gene therapy becomes very, very crucial part of the entire discussion of medical applications of a gene, medical applications of biotechnology. So that's why addition of a gene, if let's say the gene was absent, removal of a gene, the gene causing trouble, modification of a gene, again, uh, here we are talking about defective gene, and then again replacement also of a defective gene. So this is how gene therapy is largely used. Now the method, what is the method that we use, we'll discuss that later. We'll discuss about the basic process that is used in applying gene therapy. Then there is genetic testing. Now genetic testing is a kind of a medical test, right? It's a kind of a medical test that identifies the changes in chromosomes, genes, or proteins. Now, many a times, a genetic test can confirm or rule out the presence or absence of a disease, especially when it comes to the uh, genetic conditions. When you have genetic diseases, so we can actually find out if there is a genetic disease, and we can also try to find cure for it if we have been able to identify or detect the condition early. Then it will also help us in overall getting to solve that problem as well. So that's why that is done. And at the same time, there are a lot of genetic markers. There are a lot of genetic markers, meaning that there are genes which are responsible for telling you that there is a chance of onset of a disease. That sometimes presence or absence of a particular gene might give you a hint about that even if the disease does not show up right now, but there's a chance that you will develop that disease later in your life, not today. And that's why you'll see that early detection is possible many a times. You might see that today, for example, uh, there are a lot of companies which are doing home tests for you, or giving you medical packages or lab tests, full body checkups, etc., can be done. And in those full body checkups, if you have tried, you'll see that they would also say that they can do checkup for cancer, or they can do checkup for diabetes or heart conditions, etc. In many of these cases, when they take blood samples for doing these tests, they try to look for certain genes. And if these certain genes are present, then in that case it might give you a hint that it might be early stages of something that might develop later, so you can start taking precautions so that you can take care of that disease right now. So that's why genetic testing becomes very crucial, whether it is from the perspective of a genetic disease, or detection of a genetic disease, or even early detection of a lot of other diseases. So this is what genetic testing is. Then after that, we have drug administration. Now, in case of drug administration, we are saying that this is where we are making the use of biotechnology in making of drugs or medicines. So this is where we are talking about something called as biopharmaceutics. Biopharmaceutics. Now, in case of biopharmaceutics, pharmaceutics is what? Pharmaceutics is related to Pharmaceutics is related to medicines, making of medicines, or anything to do with medicines. On the other hand, bio, meaning living. So when you are making the use of living organisms, or let's say when you are combining the physical and chemical properties of the drug, the dosage form, and the manner in which it is given, to the rate and extent of drug absorption, this is called as biopharmaceutics. So biopharmaceutics tries to understand that how does a particular medicine affect the human body, whether it is having a positive impact, are there any side effects, what is the time that it takes, is it effective enough, if it is effective, how effective it is, to in what kind of conditions it is more effective. So there are all these things that we have to examine. So all these things will become a part of biopharmaceutics. So that's why biopharmaceutics examines the interrelationships of the physical and the chemical properties of the drug, the dosage form, and the manner of drug administration, on the rate and extent of drug absorption. So that's why when we try to understand what the effect of a drug is on the human body, it is called as biopharmaceutics. Now, when it comes to when it comes to uh, the biopharmaceutics, there are multiple branches of biopharmaceutics which work together in making biopharmaceutics effective, where one of the important fields is pharmacokinetics. Now, in case of pharmacokinetics, what we basically are seeing is there are two words, right? Pharma, kinetics. Pharma again related to medicines, pharma, right? And kinetics. What do we use kinetic for? I mean, where do we use this word kinetic? Where do we use the word kinetic? What is it related to? Yes, related to motion or movement. So that's why when we say kinetic energy, it is the energy due to motion. So kinetic is related to movement or motion. So when we say pharmacokinetics, it's about the motion of the medicine inside the body, that how fast does the medicine get absorbed by the body, how fast does it get excreted by the body, how fast does it start taking its effect. So when you study all these parameters as a part of biopharmaceutics, it is called as pharmacokinetics. And pharmacokinetics becomes very, very crucial as a part of this entire study of drugs and their applications on the body. Because when it comes to uh, biopharmaceutics, why pharmacokinetics becomes an important part of it also is something that you have to understand, that every disease has a different way or a different approach. That, for example, there are diseases uh, that can start showing their effect within a few hours. Correct. Within maybe 2 3 hours, 5 hours, like cholera, diarrhea, etc. They take just a couple of hours. And on the other hand, you have diseases which can take a lot of time to develop. Something like a leprosy can take up to 5 years of time to develop, right? Up to 5 years of time. So every disease has a different incubation period, the time that it takes to develop, different incubation period. Now, whenever you are making a medicine against a certain disease, you have to ensure that the drug, or let's say even if it is a vaccine, the drug or the vaccine acts faster, right? Acts faster than the incubation period. That if the incubation period is of 2 hours, the medicine should start working within an hour. If the incubation period is of 2 weeks, then the medicine has 2 weeks of time to start taking its effect. So that's why pharmacokinetics becomes very, very important, because uh, depending on how much time it takes for the incubation of the disease to happen, we actually have to plan the things out. So that's why pharmacokinetics describes what the body does to a drug, refers to the movement of the drug into, through, and out of the body, the time course of its absorption to excretion. So all these things, that's why they become a very, very crucial part of drug administration. Similarly, we have something called as pharmacogenomics. Now, pharmacogenomics, again, two words, pharma, genomics. So pharma again related to drugs, and genomics related to the study of genes, the genetic study is called as genomics. So when you are talking about the drugs and how the genetic makeup of an individual affects the individual's response to drugs is called as pharmacogenomics, meaning that genetics is deciding what effect a particular drug might have on the body. And this is also very, very important. If you look at worldwide data, you'll find that if you look at the worldwide data, you'll find that there are various different parts of the world where some kind of drug is effective and the same drug is not effective when taken in another part of the world. That a drug or a medicine being taken in Europe may not be suitable enough for a person in Asia or a person in Africa. And we have seen these instances. There have been instances where a certain medicine which was made against a particular disease is not effective in a certain genetic makeup. So that's why when we study these interrelationships, they all become a part of pharmacogenomics. So that's why pharmacogenomics analyzes how the genetic makeup affects an individual's response to drugs. Now, overall, in medical applications, one more thing is about virotherapy. Now, in case of virotherapy, we are talking about the use of virus, the use of virus and using virus as a therapeutic agent, using virus as a medical agent. So, again, in making many of the antiviral drugs, we make the use of virus. In making many vaccines, we make the use of virus. You might know about the COVID-19 vaccine also, that in COVID-19 vaccine also, various vaccines have use virus in them. Correct. So that's why whenever we make the use of virus, we say that it is virotherapy. Then, so that's, it's nothing but using virus as medical agents or therapeutic agents. Then we have recombinant insulin. Now, in case of insulin, we know that insulin is required by the diabetic patients to remove excess sugar from the blood. Because ultimately, when it comes to insulin, insulin naturally is present in our body, and it would just remove every extra bit of sugar being produced in the body. So, excess sugar can be very easily taken care of by insulin. Unfortunately, diabetic patients, in case of diabetic patients, insulin either is not made by the body or it is being made in very small amount, either not made by body or made in very small amount. So that's why they are not able to cope with all the extra sugar buildup that keeps on happening in the body, and that's why they have to take insulin injections, so that insulin artificially can be injected to the body, and it can basically start taking its effect, and it can start removing all the excess sugar buildup happening in the body. So that's why from that perspective insulin becomes important. And since insulin also is a part of the biochemical processes in the body, you can say that even insulin being made artificially is also a medical application of biotechnology. So that's why there are a lot of different ways in which biotechnology works, and we see that all these are the various aspects of use of biotechnology in medical science. Now all these points that we have discussed, all these points together can even directly become a mains question for you. They can simply ask you what are the applications of biotechnology in the healthcare sector, or what do you mean by red biotechnology, what are the applications of red biotechnology. So there are all these ways in which directly a question can be asked in your mains examination. And the smaller parts of the discussion that we have done, all these can also come in your prelims examination. The next is agricultural application, but before that, if you have any doubts, please let me know. If you want to ask something. All good. Virotherapy. So, see, in case of virotherapy, just understand the word itself. Virotherapy meaning virus being used for some therapy. So, when you are making the use of virus as a medical agent or as a therapeutic agent, like making a vaccine. In case of vaccines, many of the vaccines would use virus, correct? They use virus in making COVID-19 vaccines, as I gave you an example, or if you look at the pox vaccines also, you'll see that in all these cases, some kind of virus has been used uh, in the vaccine. So whenever we do that, it is all virotherapy. Even in making antiviral drugs, it is all virotherapy. Can you explain application in pharmacogenomics? So in case of pharmacogenomics, it is about the genes and how the genes respond to a certain drug, because not everybody in the world are not similar, because there are genetic variations all over the world. So that's why there might be certain genes which cannot which do not show any positive reaction to a drug, and they might react negatively to a drug. So that's why many a times that is to be taken into account. Or at the same time, various different genetic makeups might have different ways in which they respond to a certain drug. So that's why when you look into all that, genetics may play a very important role. That a medicine that is effective in Indian population might not be effective in the US, might not be effective in the Europe. And there are instances where we have seen it, that where the drugs which were made in a certain country, when they were exported to another country, uh, it did not have the same effect, or sometimes it actually had a negative impact rather than having a positive impact. Role of insulin in our body. Role of insulin basically is to remove excess sugar buildup from various things that we eat. The excess sugar that is built in the body, insulin would remove it and clean, you can say, clean the blood of the excess sugar. Agricultural applications, we still are to start it, we have not started it. Recently, cervical cancer treatment was in news. Yes, it was. And the vaccines also for cervical cancer have been in the news. Do we need to know how DNA and mRNA vaccines work? mRNA vaccine got Nobel Prize. Yes, we need to know how they work, and we'll discuss that when we go to the chapter on vaccines. We'll discuss about it. Right. Any other question, anyone? Anything that you want to ask? HP vaccination for cervical cancer, absolutely correct. HPV vaccine Vaccine has been in news since yesterday also. Cervical cancer has been in news since yesterday. Do you know in what context? Have you come across the news? Yeah, in the Poonam Pandey death case, where yesterday it was all circulated in the news that she passed away because of cervical cancer, and today she has come back saying that this was all a stunt. This was for raising awareness for understanding of cervical cancer and how dangerous it could be. All right. So next topic is agricultural applications of biotechnology. Now, in case of agricultural applications, do you know which crop this is? The one that you have right here on the screen. Do you know what crop it is? Yes, it is cotton. What form of cotton? BT cotton. Perfect. So this is basically BT cotton. Now, BT cotton, you can say, is one of the success stories of agricultural applications of biotechnology. BT cotton. Now, BT cotton is a cotton variant where we have mixed the genes of a bacteria with cotton. And these genes of bacteria, they actually are producing effects in a manner, they are producing effects in a manner that now these cotton plants are not getting affected by a certain pest called as bollworm. Now understand the context here. Understand the context. Cotton plants would get affected by bollworm. All right, bollworm would eat up the cotton, the flowers, and they would destroy the crops of cotton, and it would lead to a fall of up to 25 to 30% in the productivity of cotton, up to 25 to 30%. So for Indian farmers also it was a nightmare that they would lose out at least up to 25 to 30% of cotton uh, productivity or yield. Now this because of bollworm. Now this is when biotechnology was used here, and what we did was that we took genes from a bacteria. All right, we took genes from a bacteria and we mixed the genes of these bacteria with cotton. Now the genes that we took from bacteria, they were the genes that were capable of being a pesticide in itself for bollworm. So now what would happen is that now that these genes were mixed with cotton, now cotton would be able to produce its own pesticide. It would become poisonous for bollworm, and because of that, bollworm would not affect cotton. So because of BT cotton, because of this genetic modification that was made in cotton, this is what brought in almost 25 to 30% better productivity in cotton. And it really helped Indian farmers because they were able to get more out of cotton. They were now able to earn more out of cotton. So it was something that helped them increase their farm income. So that's why BT cotton is seen as one of the, you can say, success stories in case of cotton, in case of cotton cultivation. Now, when it comes to the agricultural applications of biotechnology, we are talking about something called as a transgenic plant. Transgenic. Understand this word first. Transgenic. Now, the first word trans, when do you use that term? If you think of the word like transfer. So when you say that somebody has been transferred, what do you mean? That that person now has gone from one place to another. Similarly, transport, the modes of transport, they can take you from one place to another. So that's why trans is a word that you use when you are saying, talking about a something coming from somewhere and going somewhere. So in this case, when we say transgenic, we mean by we what we mean is that we are talking about a transgene. We are talking about a transgene, which basically means a gene that is coming from some other organism. All right, transgene means that this is a gene from another organism. Now I want you to pay a very close attention to this, because I'm going to ask you a question later about transgene. So, gene from another organism. What organism? Any organism. All right. Another organism meaning any other organism. So it could be a bacteria, it could be a virus, it could be a protozoa, it could be any other microorganism, it could be another plant, it could be another animal, it could be a human, any other organism. All right. So when gene comes from another organism, we say that it is a transgene, and that's why the plants that contain these genes, they are called as transgenic plants. All right. These will be called as transgenic plants. So bt cotton is an example of a transgenic plant, because uh, it contains a gene which has come from another organism, which was a bacteria. So that's why transgenic. And in case of agricultural applications, it's largely about having these transgenic plants, or creating these plants, because they will give you a lot of desirable properties. For example, increased crop production. What we discussed the case of bt cotton, that we could increase the crop production by adding this particular gene taken from a bacteria. Correct. So that's why these are called as transgenic. So increased crop production, similarly improved nutritional value. If you look at this particular example of rice, in case of rice, there are these two samples. As you can see, this is the normal white rice that we eat. This is the normal rice. And then there is this yellow colored rice, not called as yellow rice, but called as golden rice. It is called as golden rice. Now, this golden rice is a variety of rice where you have added vitamin A to rice. Now, generally, we do not consume rice for vitamins, correct? What nutrient does rice have? What is the nutrient that rice has? Carbohydrates. Yes, because it provides energy. This is what rice does. Rice will provide you energy. So that's why carbohydrates are present in rice. So rice is a good source of carbohydrates, but not of micronutrients like vitamins and minerals. So now, for the people or for the areas where you have deficiency of vitamin A, there is another kind of rice called as golden rice, where we have artificially added genes which are responsible for synthesis of vitamin A in rice. And these genes were taken from daffodil plant. So we took the genes from daffodil plant and we inserted it in rice. We took the genes from daffodil plant. Now, daffodil plant is not something that we consume for vitamin A. There are so many fruits and vegetables that we consume for vitamin A, but they could not be used along with rice. They were not compatible with rice. Daffodil genes were. So that's why daffodil genes are used in rice. And these daffodil genes basically have something called as beta carotene. And this beta carotene helps in the synthesis of vitamin A. So that's why when these genes from daffodil is added to rice, it starts synthesizing vitamin A also, apart from carbohydrates. So that's why this golden rice is an example where we see that the genes for the synthesis of beta carotene taken from daffodil plant, and thus it helps in converting into vitamin A. So that's why wherever there are deficiencies, or if there is any disease happening due to deficiency, all that can be controlled with the help of golden rice. So it will be given specifically for areas for the people who have deficiency of vitamin A. Similarly, there can be increased shelf life. It can also have environmental benefits. So shelf life meaning that how much time can you store it for, whether it is about seeds or whether vegetables and fruits, we want longevity in it. So for getting that longevity also, sometimes genetic modification can be done. And then environmental benefits, we discussed about bt cotton. Now in case of bt cotton, what we are saying is that bt cotton makes its own pesticide. Correct. Bt cotton makes its own pesticide. What it means is that bt cotton does not need an external support. You need not use chemical pesticides whenever bt cotton is being grown. So that also means that it will help the environment also, because many of these pesticides are very poisonous, so they would negatively impact the soil, the water, the air. So all these things can be taken care of, so you can prevent at least land and water pollution. So that's why it will also have environmental benefits. So there are a lot of these benefits that we can derive out of transgenic plants. Now, when we talk about the development of a transgenic plant, what we are saying is that we are inserting a gene from another organism. Now, do we mean that this is a simple process where you take a gene from somewhere, you insert it inside the plant, and the plant starts working? Unfortunately, the things are not that simple. It's not that you can simply just take a gene from somewhere, insert it inside the plant, and yes, it starts working. That, for example, tomorrow you want your plant to start moving, start walking, then you will just need what? You will need the genes that we have, the genes that we have which are responsible for walking. Take those genes, insert it inside the plant will start moving, it will start saying, I am Groot. So all these things can happen. But unfortunately, all these things can only happen in science fiction and only happen in movies, but rather it cannot really happen in real life. Because when it comes to the compatibility, there are a lot of issues of compatibility between the genes of various plants or other organisms versus plant. So theoretically, can an animal gene be inserted inside the plant? Yes, it can be. But practically, we have seen very limited scope of that actually happening. Similarly, microorganisms, other plants, you can just take the gene from anywhere, but some might be compatible, some might not be compatible. So you first have to find that compatibility. And then there is this entire process which is called as the recombinant DNA technology. Recombinant DNA technology. So this recombinant DNA technology is the technology that is used for creating a transgenic plant. Now understand this. How exactly do we do this? What is the method that we are using in making a transgenic plant? Now, in this particular scenario, in this particular scenario, when we say recombinant DNA technology, the first step for us would be to isolate the gene of interest. We have to first isolate the gene of interest. So let's say this is the gene that we have taken from the bacteria. Let's say this is the gene that is responsible for making pesticide in the cotton plant. So first we have to take this gene. We have to isolate this gene in its pure form. So first we did that. Let's say this is the gene. All right, this is the gene that we have isolated in its pure form. Then we have to look for a vector. Vector basically means a carrier. We have to look for a vector or a carrier, which can be either a virus or bacteria. It can be either virus or bacteria. All right, so we first have to look for a vector, also called as a carrier, which can be a virus or bacteria. So largely it is virus. I mean, most of the cases you'll see that virus will be used as the vector. Rarely we see that bacteria can also be used. Now, after identification of this carrier, we have to find a suitable location where we can make a cut. We have to find a suitable location where we can make the cut and insert this gene of interest. So now this is also not very easy. It's not that you can just make a cut somewhere and just insert it inside. It's not that. It's more like a jigsaw puzzle where every piece of the puzzle can only sit at a particular place. So you will have to find that place. You'll have to find where exactly can you make that cut. So once you've found, okay, this is the place where we can actually make the cut, and that will be done with the help of a certain enzyme called as restriction enzyme. So restriction enzyme will help you identify the place where you can make the cut. Once we have identified where to make the cut, this is where we see that the cut will be made. So as you can see here, we are making the required cut. Now, once we have made the cut, now you open it. And you open it and you insert the gene of interest. So basically, this is what we have done. That now we have cut it open. So suppose that this is how it is, that we have cut it open, right? So we have cut it open, and once we have cut it open, this is where we'll see that the gene of interest will be inserted. So this is the required gene, this is the required gene, and now we insert the gene of interest in it. So this is the gene of interest, this will be inserted right here. So this gene of interest will now be inserted like this, right? The gene of interest is now inserted, and once you have inserted the gene of interest, then comes another enzyme. So first one was restriction enzyme. So restriction enzyme helped us in identification. Restriction enzyme helped us in identification of where to make the cut. All right, identification of where to make the cut, and it makes the cut. And then after that, once you have inserted the required gene, then comes another enzyme called as DNA ligase. And this DNA ligase is like, you can say, Fevicol. All right, it's like a Fevicol or a Feviquick, which is making sure that these two ends can be made to stick together. So that's why you have this DNA ligase. We have this DNA ligase, and then you have this. This basically is the gene of interest. This is the gene of interest. And then after that, this is where you have the vector gene. So this is how all these are now combined together. And once these are all combined together, this is where now this entire DNA is called as the recombinant DNA, where you have a combination of two different pieces of genes. This is called as the recombinant DNA, and hence this technology has been named as the recombinant DNA technology. Now, once we have produced this recombinant DNA, we produce multiple copies of it. We start doing photocopies. In a manner, we start doing photocopies. So we produce multiple replicas. We produce thousands to lakhs of replicas of this recombinant DNA, and then finally it is inserted inside the plant. So unfortunately, that's why it's not a simple process where you just catch hold of a gene and insert inside the plant. First you take the gene of interest, then accordingly you have to find a vector and find the place where the cut can be made in the vector with the help of restriction enzymes. Once the cut has been made, you insert the gene of interest, you stick the ends together with the help of DNA ligase, which is like a glue, which is like a Fevicol or a Feviquick, and then after that you create multiple copies of it. And this process of creating multiple copies is called as PCR, or polymerase chain reaction. And then finally you insert this recombinant DNA inside the plant. So this is the entire process that is used in making of the recombinant DNA, or the entire process of recombinant DNA technology. So first we start with the isolation of a genetic material, correct? So first isolation of the genetic material, where you would isolate the desired DNA in its pure form, free from any other macromolecules. And then after that, you have to make the cut. So for that we need restriction enzymes, and these restriction enzymes will play a major role in determining the location at which the desired gene is to be inserted. And then after that we have DNA ligase. So DNA ligase will be, and this is an enzyme called as DNA ligase, and this is something that will help in joining of the two pieces together, the gene of interest and the vector. And then after that we have polymerase chain reaction. So polymerase chain reaction is basically amplifying of a single copy of DNA into thousands to millions of copies, meaning that now we are creating multiple copies of this DNA, and this process is called as PCR, or polymerase chain reaction. And once we have created multiple such copies, This is where ultimately this recombinant DNA is introduced to the recipient host cell, and even this process has a name called as transformation. So ultimately, this is the entire set of steps. These are the entire set of steps which leads to the formation of a transgenic plant. All right, so this is how you get the transgenic plant at the end of all these steps. Read this question. I'm launching the poll for you. Please mark your answers. Hmm. So 67% of you have marked the correct answer. Let me just discuss this with you. Recombinant DNA technology or genetic engineering allows genes to be transferred across different species of plants. Can we do this? Yes, we can do this. From animals to plants, can we do this? Yes, we can do this. From microorganisms to higher organisms, can we do this? Yes, or yes, we can do it. What did we discuss? You take it from any organism. You can take it from any organism. There is no restriction. There is no restriction where you can't take from. So that's why it does allow the genes to be transferred across the species, whether from plant to plant or animal to plant or microorganism to plant or a human to plant or an alien to plant. Everything, everything. All right, so that's why it does allow everything. So that's why just remember that there are uh, all these can be done. So it's not that there is any restriction. So that's why the correct answer will be 1, 2 and 3, that it can be done in for any organism. That's why just remember this, any organism. So it does allow things to happen from even from plants to animals also. It has been done. There are experiments we have seen, plants to animals, gene transfer also. So yes, it is all possible. Now, coming to some of the other applications of biotechnology, we have agricultural applications where we understand about biofuels. Where in case of biofuels, we also had a discussion where we understood that biofuels is where you are also biofuels is where you are actually using crop plants or edible crops, right? We discussed about the first generation biofuel, if you remember, where we discussed that edible crops are used. So that's why in case of biofuel also you can call it as an agricultural application of biotechnology, because this is exactly what you're doing there, that you are using crops or living organisms for some kind of application. So that's why biofuels can also be called as agricultural application of biotechnology. Similarly, Similarly, when it comes to uh, other applications, we have something called as biofortification. Now, in case of biofortification, here we are talking about uh, a method in which we are increasing the nutritional value inside a plant, increasing the nutritional value. Now, understand how to use or how to increase the nutritional value. We discussed about golden rice, that in case of golden rice what we are doing is that we are enhancing the amount of vitamin A, or rather we are adding vitamin A to rice. So in that case it will enhance the nutrition or the nutrient value of that crop. So that's why development of micronutrient dense staple crop variant using the best traditional breeding practices and modern biotechnology is called as biofortification. Now, micronutrient. Do you know what is micronutrient? What do we mean by micronutrient? Any idea? What do we mean by micronutrient? Yes, required in less quantity. The nutrients that our body requires in less quantity, exactly vitamins and minerals. So these are nutrients, nutrients required in less quantity. Nutrients required in less quantity, that is basically vitamins and minerals. Vitamins and minerals. So that's why these are called as micronutrients. On the other hand, you have macronutrients where you have carbohydrates, fats, even proteins, for example. So these are called as macronutrients because they are required for in the body in larger quantities. So, for example, I mean, if you go to a nutritionist and they would talk, and let's say you tell them that you want to lose weight, and you go to them, so they will tell you that you have to cut on your macros. They'll always use the term macros. So macros is nothing but the macronutrients because they are stored in the body also in larger quantity, because body makes sure that they have that it has enough of energy, that tomorrow if you do not provide that energy, they'll already have the store of it. Whereas uh, for day to day activities, micronutrients do not play that role, uh, the role that is played by carbohydrates. So body always craves for carbohydrates and wants more of carbohydrates. It will keep on storing all the fats and carbohydrates, but will always need that extra amount. So that's why macronutrients. So that's why fortification is not really required in the macronutrients, it is largely required in the micronutrients, that even if there is deficiency, the body still works. And maybe you can take supplements, maybe you can eat those fruits and vegetables which are required for these micronutrients, and then you can re-establish them in the body. But in case of macronutrients being lost, it could be a very different scenario altogether. So that's why vitamins and minerals, they are called as micronutrients. And when through plants or through your food, if you are using the process of uh, inserting these vitamins and minerals inside the crop, then in that case it is biofortification. But in general, fortification is addition of an ingredient to the food. That even, for example, let's say iodized salt. Iodized salt is something that probably every one of us are using at homes. So iodized salt is an example of fortified food, but not biofortified food. Biofortification is when you are adding to the crops, then it is biofortification. Otherwise, it is normal fortification. So that's why iodized salt is an example of fortification. Golden rice is an example of biofortification. I'll write it. Iodized salt is fortification, whereas golden rice is biofortification. And then there is a related process called as supplementation. When you take supplements, you take medicines like vitamin D tablets or vitamin B12 tablets, etc., then it is called as supplementation. And then after that, there is an issue that WHO talks about called as hidden hunger. Now, hidden hunger is a concept where we are saying that although you are eating enough, but you are not getting all the nutrients that is required. So maybe you are getting enough of carbohydrates, but you are not getting all the vitamins, all the minerals that are required as a part of somebody's balanced diet. Now, this is a bigger problem for countries which import food, where they are not agriculturally capable, and hence they are importing fruit from somewhere. Now, whenever the imports happen, the imports are largely about rice, pulses, etc. So they might be less in micronutrients and have enough of carbohydrates, etc. So then in that case it is called as hidden hunger, that even though you are eating enough, but you're not getting all the nutrients that are required for healthy living. So that is where we see that this becomes a problem. And WHO has been talking about this problem, where they are saying that this is a form of undernutrition, where although the quality or the quantity that you are eating is enough, but the quality of the food that you're eating is not enough, and hence you're not getting all the nutrients that are necessary. So that's why this is where quality of food quality of food is not adequate. The quality of food is not adequate to meet but instead of adequate, let me say enough. That it's not enough to meet the nutritional requirements, to meet all the nutritional requirements. So this is called as hidden hunger. Now, overall, there have been a lot of examples of biofortified food. We have seen that it has been used in all these various ways. So, whether it is cassava or maize or sweet potato, in all these cases, we have seen that it has been used in one way or the other. Now, if you, for example, look at cassava, so cassava is where we have seen that vitamin A fortification has been done, even zinc fortification has been done, iron fortification has been done. So there are various fortifications that has been done. Similarly, in case of maize also we have seen that there are various fortification that has been done. So for example, let's say especially in case of maize, we have seen that vitamin A, here also vitamin A, and there is a case of also selenium infusion. So selenium additions also have been done in maize. Similarly, when you look at sweet potato, so in case of sweet potato also we have seen that vitamin A fortification has been done. Then if you come to beans, so then in case of beans also we have seen that largely we have done iron fortification. Largely we have done iron fortification in this. And then in case of pearl millets, there are two fortifications that we have seen. One has been about iron, so we have done iron fortification, and we also have done calcium fortification in pearl millets. And then in case of rice and wheat, we have seen vitamin A fortification being done, and at the same time even zinc fortification is something that we have done in case of rice and wheat. So that's why we see that there are all these cases, all these ways in which we have been doing fortification for various different crops. So there are all these things, all these examples that we have of fortification. Then we have also seen that genetically modified crops can also be used as animal feed. They can be used as animal feed. So using them as animal feed means that we are talking about genetically modified crops which can be used as food by animals. And here what we want is a good nutrition for the animals, right? Because animals, we need not have animals consume a lot, but with limited amount of food, if you can give them all the nutrition that is required for their functioning, that's what also can be done with the help of genetic modification. Similarly, we have pest resistant cultivars. We discussed about the bt cotton case, where we know that they will help in uh, making them pest resistant. Similarly, there are HT variant crops or herbicide tolerant crops. Now, herbicide tolerant crops are the kinds of crops where we see that they reduce the production cost and help in the weed management. Because what happens is that these crops will not be negatively impacted, all right? These will not be negatively impacted by the herbicides. Now, what are herbicides? Herbicides are the chemicals that you use against weeds, to kill the weeds, the unwanted growth that is happening in the field. So, when you want to kill the weeds, you use herbicides, and sometimes these herbicides can have a negative impact. All right, the herbicides can have a negative impact on the crops also. So that's why when you have the HT variant crops, they will not be affected by the herbicides, and the herbicides will be able to clear off the entire agricultural field. So that is also where we can make the use of them. Then the use of biofertilizers. Now, we discussed that biofertilizers are also produced out of some of these processes. Like even in biogas production, we discussed that biofertilizer is a byproduct. So using biofertilizer is also an example of agricultural application, because there also you are getting it from the agricultural processes. So that's why we see that the use of biofertilizer also is one of those examples. And then improvement in floriculture also. So it's not about only the food crops, but also in the commercial aspects of agriculture also, where we see that biotechnology plays a major role. And this is where we see that in floriculture it can play, or it rather plays a major role, because every requirement that you have out of a flower, that you would want a good fragrance, you would want very good colors or good contrast, and maybe you want a bigger size, and at the same time you also want them to live longer and do not they should not die in a couple of days. So in all these cases, to ensure all these things, genetic modification again can be used. Then apart from that, there have been some other cases also, like the case of GM mustard, for example, which has been in the news. We'll discuss that later, about what is GM mustard and what are the issues, etc. Then apart from GM mustard, GM rubber also has been in the news, where there is a genetic modification in rubber that we have tried in India, so that rubber can be grown in the northeast, especially in the states of Assam and Arunachal Pradesh. So that's why GM rubber also has been in the news. So overall, we see that there are multiple different ways, there are multiple applications that we see of biotechnology in terms of the use that we see of biotechnology. Read this question. All right, if you have read it, I am launching the poll for you. Well, let me not launch the poll for you. In this question, there's a reason why I'm not doing that. Please tell me about what answers do you think is the correct answer in the comments. A lot of C's and a lot of D's. Hmm. So let's discuss this question. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? First is to enable them to withstand drought. This is something that we have been doing, that how can we grow these plants in low water areas, in water deficit areas. So it's possible when we have done it. So this is correct. To increase the nutritive value of the produce. This is also correct. We discussed about this as well in case of golden rice. Statement number 4, to increase their shelf life. We discussed about this also, that for seeds and for the fruits and vegetables also this can be done and has been done. So 1, 2, and 4 definitely correct. Statement 3, to enable them to grow and do photosynthesis in spaceships and space stations. Now this is where all the problems begin. because to enable them to grow and do photosynthesis in spaceships and space stations is now possible, has now been demonstrated. There have been multiple experiments that NASA and other space agencies have conducted, and they have shown that it is possible. I think it was 2015 when the first time they had showed how to grow lettuce in International Space Station. So already done. So now already done, after 2015 already done. So now it is correct, but in 2011 it was not. Because look at this question that we are talking about: what are the prospects for which the plants have been created? Now it's not a can be question. Another way to ask the same question could have been can be, that what are the prospects for which genetically modified crops, genetically engineered plants can be created. Now can be anything. So when if it was about can be, then you can simply close your eyes and mark all of the above. But when it says have been created, it simply means that unless you have an example, that unless it has already been done, then only you can mark it as correct, otherwise not. So that's why the language of the question matters. It becomes very very important for you to read the question carefully in many of these cases. So that's why when we look at this question from the perspective of 2011, the answer should be C. And when you look at the same question from the perspective of 2024, the answer has to be D. So this is how the world changes. So that's why I did not launch the poll, because there are two answers which are correct depending on how are you approaching the question. Thinking from the perspective of when the question was asked, or thinking from the perspective of today and what we have seen with all these uh, aspects of biotechnology in today's world. Sorry, this is repeated. Then after this, we have white biotechnology. Now, white biotechnology is basically about industries and how we can use biotechnology for various different industrial processes. For example, the process of fermentation, which produces alcohols, and alcohols are used as industrial reagents in a lot of different industries, in a lot of different applications. So that's why that becomes an industrial application when microorganisms etc are used for the process of fermentation. Similarly, in making preservatives, industrial process. Biofuels, again an industrial process, because in the industries you are producing it industrially, these are being used. So that's why all these can be counted as white biotechnology. Then apart from these basic processes, there are some specific processes also. For example, there is something called as biosorption. Now, biosorption, when you break down the word in a manner, we are saying we are talking about biological absorption. So that's why the term is biosorption, biological absorption. So now this is the property of certain types of inactive, dead, or microbial biomass to bind and concentrate heavy metals from even very dilute aqueous solutions. Where we are seeing that even where the concentration of these metals are very low, even from there we can extract them. And largely we are talking about pollutant extraction, these heavy metals which can be toxic. So we are talking about removal of the toxic elements, we are talking about the removal of the toxins from the environment with the help of microbes. So what may happen is that these microbes can go to a dilute solution or a dilute environment, and they can start consuming or absorbing these heavy metals. Although for microorganisms to do that is also very very difficult. It's not that they can completely decompose the heavy metals, because the heavy metals are uh, structures which are not easy to break down, and that's why microbes may not be able to do that. They may not be able to break down but absorb it. That's why biosorption, they do not break down, they do not biodegrade, they absorb. Understand the difference, that they absorb and they do not degrade. So that's why microorganisms can be used in pulling out all the heavy metals or the toxins from these given environments. Then there is this process called as biomining. Now, in case of biomining, again, we are talking about biological mining, where you are making the use of biotechnology or any biological agent for mining purposes. Now, here also, whether it is about petroleum mining, whether it is about metal mining, in all these cases, uh, plants or even microorganisms can be very, very helpful. So, we have seen that we can actually derive help from all these. So let's say, for example, microorganisms, they can be used to leach out minerals, and that's why they would be able to help, especially when it comes to, let's say, whether it is about uh, any petroleum product or any metal for that matter, aluminium, copper. In all these cases also, microorganisms can be useful because they can help in extraction of all these, and especially when it comes to, let's say, dilute areas where the concentration is very low, then using the industrial process is not advisable, because you'll be spending a lot on providing the energy to that entire process and getting a lot less out of it. So that's why in these cases, using microorganisms is a better alternative than using the industrial processes. Similarly, even plants can be used, and this is called as phytomining. Phyto, just remember that whenever you come across this term phyto, the meaning of the term phyto is plants. Right? Phyto means plant. So wherever we are making the use of plants, the term phyto can be used. Now here the roots of the plants can be used. We can make the use of the roots of the plants, and these roots of the plants can absorb all the metals from the soil, and then you can just take away the roots of the plants, burn it, and from the ashes of the root you can actually collect the metal. So there are things like these also that we do. There are these processes that we use in case of biomining. So that also becomes a concept related to biotechnology. So that's why in the industrial processes, we do see that there are a lot of ways in which we can make the use of biotechnology. Similarly, when it comes to blue biotechnology, we are talking about biotechnology in aquatic environments. So whether it is about growth of some underwater plants and animals, or even the growth of algae. If you remember, we discussed about algae based biofuel. We discussed about algae based biofuel. So algae based biofuel also becomes an example of blue biotechnology. Similarly, cleaning up of the ocean water or cleaning up of the sea water with the help of biological agents or microorganisms, that also can be called as blue biotechnology. For example, let's say in cases of oil spills. Now, in case of oil spills, what happens? That oil will just form a layer on water, correct? Oil will form a layer on the surface of water. This is where we use a bacterial formulation called as oil zapper. Now, this oil zapper is made of 5 different types of bacteria, and all these bacteria are mixed together, and they would be taken to the place wherever oil spill has happened. And then these bacteria will start eating up on the hydrocarbons that you have in that oil spill, and they would break down the hydrocarbons into carbon dioxide and moisture, meaning carbon dioxide and water. So hydrocarbon will break down into carbon dioxide and water with the help of these microorganisms. All right. The bacteria, rather. So that's why this oil zapper becomes a very effective technique in treatment of oil spills in the ocean or in the seas. So that's why there are all these ways in which we can also make the use of blue biotechnology. Now, before going to the environment, if you have any questions related to whatever we have discussed until now, any unanswered question, from where the gene of interest comes. So gene of interest can come from anywhere. For example, in case of bt cotton, the gene of interest was taken from a bacteria, correct? The bacillus thuringiensis bacteria. So we took the gene of interest from bacteria. In case of golden rice, we took the gene from a daffodil plant. So in that case, the gene of interest came from daffodil plant. So the gene of interest can come from any organism. but this genetically modified cannot change DNA of animals while consuming them. uh, I don't exactly understand what your question is, but if I'm correct, you are talking about whether they can make genetic changes. So, see, genetic mutations sometimes is seen to be possible. There are various papers which have been published that due to a biological agent, there are chances that genetic mutations could be possible. So, the answer could be the answer is basically that there are chances that something like this may happen. Use of biotechnology. What is this question? This entire chapter is use of biotechnology. We discussed about all the medical applications, and now then we discussed about all the agricultural applications. Then we discussed the industrial applications. Then we discussed the use in water, and next up after the break would be about the use in the environment. So this entire chapter is nothing but the use of biotechnology. Why would you ask this question? After the use of microorganism, it would die or not? The use of microorganisms, it depends on what you are using the microorganism for. I mean, if you are extracting the gene out of microorganism, uh, if it is a very crucial gene out of it, you may not be able to make it survive. Or if, let's say, it's a process of the growth and development, then in that case it would survive. Is it possible that COVID was leaked bioweapon of China? It is perfectly possible. We'll discuss that about the problems of biotechnology later. Biotech includes all kinds of activities involving living agents, or is it only at gene level? No, everything involving living agents, you can say that it is biotechnology, not necessarily only gene. As far as I understand, fortification and biofortification is not always done at gene level. That's correct, 100% correct. For example, iodized salt. So yes, that's what I told you, that fortification, iodized salt is an example of fortification. Whereas if, let's say, even a hybrid variety, if you produce a hybrid variety, you do not make any genetic changes there. You just cross pollinate or hybridize two varieties of plant and they might work. And then in that case, there is no genetic modification or genetic engineering hasn't been used there. So that's why it necessarily need not always mean that biotechnology is the use of genes. As I'd given the example of curd, if you remember, that curd is an example of biotechnology, but not the use of genes in that. Mosquito gene is modified. I mean, mosquito gene modification is possible. There is a method called as the Wolbachia method that we use in modifying the genes of mosquitoes so that they may not be able to let the virus grow in their body. Your way of teaching is too good. Thank you, Shiv, for the compliment. What is plasmid? So plasmid, basically, you can say in a manner, these are genetic materials which are found outside the chromosomes, especially when you have smaller microorganisms, or let's say simpler microorganisms. In them we see that they have plasmids instead of having a very well structured nucleus and all the chromosomes being inside that nucleus and everything being well arranged. So whenever the things are not well arranged, this is where we see that plasmids will be found. Right, any other question, anyone? All right. So let's take a break here. I think part three is what we have next. So in part three, we'll start with the use of biotechnology in the environment, bioremediation, and then after that, some of the very important concepts like stem cells and everything related to stem cells. What is cloning? What is DNA fingerprinting? The issue with GM mustard, the concept of three parents baby, and then all the problems related to biotechnology in terms of dark biotechnology. So we'll discuss about all these concepts on the other side after the break. So we'll come back at 6 o'clock. I think 6 o'clock is when you have the schedule of your part three. So let's come back at 6 p.m. and continue with our discussion.